So now we will talk about cell division. Why is cell division important? Cell division is important as it forms the basis of our life. For various processes such as repair of our damaged bodies or for reproduction, cell division is required. So the cell division is of two types, mitosis and meiosis. The difference between mitosis and meiosis lies in the separation of the genetic material. In mitosis, the genetic material or the number of chromosomes is equal in daughter cells as in the parent cell. However, in meiosis, the genetic material is halved in the daughter cells. So, the mitosis is required for asexual reproduction or for the formation of tissues, etc. However, meiosis is required for sexual reproduction. So, today we will study about mitosis. Mitosis consists of two phases, interphase and M phase. The interphase itself consists of three phases, G1 or the gap 1 phase, S or the synthetic phase, G2 or the gap 2 phase. So during the gap 1 and the gap 2 phases, various processes occur such as formation of RNA and during the gap 2 phase, repair of the damaged genetic material occurs. However, the most important phase in interphase is the S phase or the synthetic phase. During the synthetic phase, the replication of the genetic material, which is the DNA, occurs. So the interphase forms occurs for about 90%, 95% of the time of mitosis. So it is a very it takes a lot of time. And after the interphase comes the M phase. M phase consists of karyokinesis and cytokinesis. So karyokinesis is the division of the nuclear material, whereas cytokinesis is the division of the cytoplasmic material. So first we will study about the karyokinesis. Karyokinesis consists of four phases: prophase, metaphase, anaphase, and telophase. So the first one is the prophase. During the prophase, the condensation of genetic material occurs, formation of spindle fiber occurs, and the two centrioles migrate towards opposite sides of the cell. After the prophase comes the metaphase. During the metaphase, the sister chromatins align themselves towards the equator and they are perpendicular to the spindle fibers, as we can see in this. After the metaphase comes the anaphase. During the anaphase, the sister chromatins are pulled towards opposite sides of the cell, as we can see. After the anaphase comes the telophase. During the telophase, the nuclear membrane is formed again, and the genetic material again is again reorganized. As in prophase, as we can see, the genetic material was organized by dehydration. It was condensed by dehydration, but in the last phase of karyokinesis, which is the telophase, again the genetic material is decondensed. So this was the division of the nuclear material. After the division of the nuclear material or the karyokinesis, then we come to cytokinesis. So the cytokinesis in animal cells starts at metaphase, during which the cell organelles start organizing themselves at opposite sides of the cell. So in animal cells, the cytokinesis starts by formation of a cell furrow, as we can see here. So after the cell furrow is formed, the division of the cytoplasmic material occurs, and here we have two daughter cells. So our mitosis is over. So as we can see, this was our mitosis, and we will talk about meiosis next.